Awesome. Take it away.
All right. Well, thanks. This is very exciting. I think this is a very fun initiative. So I'm very happy to be here. What I'm going to present on today is a work that I did as a postdoc at the University of Tennessee. So first of all, I'd like to acknowledge that the university operates on traditional lands of indigenous peoples, including the Chippewa, the Yuchi, and the Cherokee. Mm, There. So what you see on your screen is a white-tailed deer being swabbed to test for SARS-CoV-2, the virus that causes the COVID-19 disease. And this white-tailed deer are one of the most notable examples of this virus being found in wildlife. There are many reports that it's widespread in many different populations. There's evidence that they acquired it from humans, that they can transmit it to other deers, and there's also evidence that they can also transmit it back to humans. And to me, this is one of the most compelling examples of why we need to understand spatial ecology in order to understand disease transmission, especially for wildlife. There needs to be some spatial and temporal overlap between individuals or between species for there to be this spillover or these transmission across populations. And this is what we have tried to do, to provide a framework to use spatial information and specifically movement data to understand how disease is transmitted, and especially in wildlife. And the rationale for the method that we've developed is fairly simple. Like if you imagine two individuals as shown here, one red and one blue, and this can represent anything from elephants to mice to salamanders, as long as they move across the landscape. And you can obtain some information about how they are moving. Now we have more and more methods available to determine like these very detailed, these very fine scale tracks of how animals move. And when you have that information, when you have that history, you can estimate or you can know where those individuals interacted directly. That is, they were at the same location at the same time, but also where they interacted indirectly. That means where their tracks cross, which is shown here in the black circles. What this also allows you, having this detailed information, is you can use this to build animal movement models, and from that, understand how these animals are using the space. And there are many methods developed, and especially in the animal movement ecology literature, that allows you to extrapolate this to the scale of the landscape, which is something that we took advantage of for the method that I'm going to explain. And the formulation, well, yeah, essentially what we want to know from these data is what is the force of infection, and that is defined as um, the risk of transmission that one host experiences due to another, and we want to estimate that across space and across time. So I'll be referring to this as the FOI. The formulation, conceptually, it's fairly simple. If you take one of those individuals that I just showed you at some point along those tracks, and you want to know at a certain point whether the other individual was there at the same location at some past time, if it was there, whether it was infected, and how often it would shed propagules of some infection stage of the parasite. And based on the time that had passed since the other host was there, whether it would be likely that those propagules would have survived. And you ask this for like all possible past times, integrate that, and you obtained the force of infection for that particular location, for that particular pair. And that's something that Mark Wilbur, my supervisor, developed in a paper in 2022. This is the basic equation, but what I would like you to focus on is this second equation, which is what we have developed. There is one limitation, or one caveat to the original formulation, and it is that you're kind of limited to what you observed. You can calculate this force of infection for the specific locations where the animals were, but no other locations. However, as I said, with animal movement data, you can obtain a probabilistic expression of where animals could be. So we take advantage of that and calculate instead an expected force of infection based on the probability that animals will be at a certain location at any point in time. Now, there is one wrinkle to this, and it is that when you have those, they're called utilization distributions, which is the probability that animals will use some locations in space. That doesn't necessarily mean that you can estimate directly the probability that will come into contact with each other. To actually have this, if movement is not independent, you need to account for the correlation in space use between individuals. And that's what's shown in the equation in the bottom, sorry. So what this equation shows, and this is the main equation of our approach, is that the force of infection here depends, first of all, on the shared space use, which is shown here by the terms PI and PJ, which are those utilization distributions I mentioned. This is also obtained, as I will explain, from the utilization distributions. But very importantly, you need some correlation, some metric of the correlation in space use between the individuals. And I'll explain more in a minute. The process is as follows. You start off with some movement paths, shown here in red and turquoise. And these are very similar by design because these individuals are attracted to each other. From this, you build a movement model, and you can extrapolate this across space to build what's called the utilization distributions. And again, these are very similar because the movement is very similar. But instead of having just these lines, you obtain probabilities across space. And from this, you obtain two terms in the equation that I showed you before. First, here is the product of the UDs themselves, which is in a way an estimation of the probability that they would come into contact with each other if movement was independent. And then secondly, you obtain the product of the standard deviations in those utilization distributions, which is a metric of how variable that estimation can be. Now, in parallel, you can estimate from these movement paths for every location in space what's called a location history. So for every grid in space, you determine whether each individual was there or not for every point in time. And you can calculate at every cell the correlation between those position histories. So it's a correlation between two vectors of zeros and ones. And what you do to account for the lag or the past times, which is the key element here, is that you do the lag correlation. So you calculate the correlation with lag 0, and then 1 time step back, and then 2 time steps back, and then you do that for all possible, or a lot of the possible lags. And this gives you this integrated correlation term, which is much more heterogeneous in space. You combine these three and obtain the force of infection across space. So here, you see that it's also very heterogeneous in space, but it's related to that correlation term, which is critical. So we wanted to know, having that framework, how that estimation of FOI is influenced by different biological parameters and different movement parameters. So we simulated some tracks, a lot of tracks, and then changed that interaction term that I told you about before, and that's shown here on the x-axis. So an interaction strength of 0 means that the two individuals in the pair are moving completely independently from each other, and then 1 means that their tracks are completely identical. And what I would like to highlight here is that these values in red are what you would estimate when you take into account that correlation in space use, which is orders of magnitude larger than the blue one, which is the estimation if you assumed that they were moving independently. And this is very important when you're trying to estimate the risk of transmission, because you can't always assume that animals are moving independently. There is another important result that we found, and it is that this ratio between these two results is highly dependent on the biology of the parasite, and more specifically on the how long it survives in the environment. Parasites that survive very short periods of time, so here close to 0, For those parasites, that correlation, the importance of that correlation is much greater than if you see here when the time to decay is much longer. And this has very real implications for actual empirical systems. So we actually tested this on an empirical data set of 5 white-tailed deer that were tracked in Tennessee. We did the same process. We calculated the FOI across space for all pairs. And what I would like you to focus on is on the bottom here. When we have more than 2 individuals, we can actually calculate or build networks and estimate metrics like R naught based on those contact networks. And if you focus here, this is the network that was built and the R naught that was calculated when we ignored the correlation term. So if we assume that all animals were moving independently, and you can see that the relative R naught is 10 times greater when we actually account for the correlation, which in this case, in the case of the deer, is probably related to social interactions between females. And this is something that, again, should be taken into account very importantly for monitoring wildlife disease. So something that we like about this method is that we have shown that for some diseases where the parasite lives or persists long in the environment, you can actually estimate or have a very good approximation of the expected FOI across space, across the landscape, based solely on the utilization distributions when you can assume safely that movement is independent between individuals. When it is not, however, and this is an outstanding challenge, we need to have a way to estimate the expected correlation, not only based, because right now it's only based on the observed data, but if there was a way to properly estimate how to calculate the expected correlation and link that with environmental variables and with social interactions, then our method would be that much more powerful. So yeah, it's some very exciting challenges. I'd like to thank you for listening, and I'm happy to take any questions.